Dear colleagues, my name is Valentina Nekljudova. I am a statistician at the German Press Group, and it is my honor and my pleasure to give a talk at this year's San Antonio Breast Cancer Symposium about how to design an efficient clinical study. I have nothing to disclose. The topic of the talk is very broad. Since I am a statistician and the majority of the audience probably consists of clinicians, choosing material for a 20-minute talk, I concentrated on the aspects of the clinical trial design, requiring a lot of interaction between these two professional groups. Here's the plan of today's talk. We will start with a very basic, very simplified idea of how a clinical question can be translated into the study design. Then I will introduce the language of estimates, and we'll talk about some attributes of the estimates, mainly about endpoints and intercurrent events. And we will move to statistics, sample size, alpha spending, and interim analysis. The aim of a clinical trial is always to answer a clinical question. How does a typical clinical question look like? Usually it is whether one treatment is better than another for some type of disease. I argue that a lot of trial design is already in the question. Let's explore how much, and to make this exercise less boring, let's switch for a moment from diseases to cats. We all know that cats love to sit in the boxes, but does the color of the box matter to a cat? Let's ask the question, are yellow boxes more attractive for cats than red ones? What is already clear from the question? Who is this question about? It is about cats. We will need cats for our potential trial. What are the treatments we are going to apply to them? These are yellow or red boxes. The next point is, how are we going to measure attractive in a single cat? I suggest that if cat goes to sit in the box, when offered a box, the box is attractive. The last point is, how do we aggregate single observations in the measurement of more attractive? At this last step, the real statistical methods usually come in. Let's not invite them in yet and try to imagine how a child with no knowledge of statistics would design a trial about cats. So let's imagine that a kid has 8 cats, 4 red boxes, 4 yellow boxes. She has asked herself whether the color of the box matters and wants an immediate answer. What would she do? She could offer 4 yellow boxes to 4 of her cats, 4 red boxes to other 4 cats, and then see and count what happens. Let's see. Yellow. The first cat goes in, the second one goes in, the third one ignores the box, the fourth one goes and sits in the box. Now the red, the first one likes the box, the second one doesn't, the third one likes the box very much, and the fourth has better things to do. Let's count. 3 cats, or 75% of cats offered yellow boxes, accepted them, and 2 cats, or 50% of cats offered red boxes, like them. So we get 1 cat, or 25% difference in favor of yellow. Now our kid has her quick answer. For us, this trial is too primitive and too small to be able to generalize from 8 cats to all cats in the world, but it is still suitable to illustrate the most important parts of a trial design. Now let's have a look how they are called in the estimates language, which gives structure and precision to what we do designing a trial. There will be a literature list on one of the last slides. ICH E9 guidance, statistical principles of clinical trials, received an addendum R1 2 years ago, which is called Addendum on Estimates and Sensitivity Analysis in Clinical Trials. It suggests to think about every statistical analysis of any trial objective in terms of estimates. An estimate consists of 5 parts or attributes, 4 of which our imaginary child has already kind of guessed. You can think about an estimate as of a checklist of important things to define as precisely as possible when planning a trial. The first one is population. These were 8 cats in our cat trial, or this would be patients enrolled in the trial according to the inclusion criteria, or a subgroup if we are talking about an estimate for a subgroup analysis. Defining a population for your clinical trial, you have to find a balance between including a group of patients who have a real unmet medical need, but not restricting the group too much and keep it large enough to be able to feed your trial. The second attribute of an estimate is the treatment of interest and alternative treatment. These were yellow and red boxes in our cat trial. In the real trial, this would be experimental drug versus placebo or control. Nowadays, when standard of care is getting more and more versatile, the choice of control arm can get pretty complicated and should be thought through thoroughly. The third attribute of an estimate is endpoint, and how do we measure it in a patient. In our cat trial, it was the cat going into the box, and I will talk about endpoints for breast cancer trials in some detail on the next slide. The fourth attribute of an estimate is the population level summary. This was difference in box acceptance by color in our cat trial. In real trials, here the statistical analysis takes place. Please note that it doesn't work if the first 3 are not right. You cannot save your trial with fancy statistical methods if you made bad choices for the first 3 attributes of your estimate. There is one more which our hypothetical child hasn't thought about for the cat trial, which is intercurrent events, and I will come back to them a little bit later. No trial stands alone. There is a shared context of clinical knowledge, and to be able really to contribute to this context, the trial data must be comparable with the data of other trials investigating the same or similar questions. To achieve this, standardized endpoints are very helpful. Here are some for breast cancer and where to find them. For adjuvant breast cancer trials, there is STEEP, which stands for Standardized Efficacy Endpoint in Clinical Trials. STEEP was first published in 2007 and was updated this year. It describes, for example, invasive disease-free survival, distant disease-free survival, and overall survival. In the table below, you can see what is considered as an event for each of these endpoints. Invasive breast cancer-free survival was added with this year update and differs from the IDFS in not counting the secondary malignancies as an event. I refer to the publication for the discussion of the implication to be considered choosing one or another. PCR can be used for accelerated approval for neoadjuvant trials and for the regulatory purposes for the neoadjuvant trials. The time to event endpoint of choice is event-free survival, which includes additionally the non-operable progressions under neoadjuvant chemotherapy or progressions requiring non-protocol therapy before surgery. For the sake of completeness, I also listed some for metastatic breast cancer with a reference. Now let's move to the intercurrent events. which are not a new aspect, but they are very much in focus of the E9 addendum. Here's the definition. Intercurrent events are events occurring after treatment initiation that affect either the interpretation or the existence of the measurements associated with the clinical question of interest. Here are some examples. How do we deal with intercurrent events? The E9 addendum suggests several strategies. I picked up two which I consider most important for breast cancer trials. The first one is the treatment policy strategy, which is just to ignore them. Compare it to the ITT principle, to the intention to treat principle we are all familiar with. The problem is that it is not always possible. It is easy to ignore discontinuations, but you cannot ignore, for example, deaths. And for deaths, another strategy, which is called composite variable strategy, is better, where you build the intercurrent events into your endpoint. For example, you can consider patients with no response measurement due to an intercurrent event as no response, or you define survival instead of time to event endpoint, where you count death irrespective of any cause as an event for the endpoint. This is what STEEP is doing. Statisticians have competing risk methods in their toolbox, which can deal with intercurrent events. As a clinician, you must be very clear on the interpretation of this kind of analysis. The most important point here is that sensitivity analysis may be needed and are recommended in cases where there is no black or white best way. For example, including both IDFS and IBCFS in the trial would be a sensitivity analysis, and STEEP update recommends to collect data for both and report both. You will have, of course, to define which one is your primary analysis, and the sensitivity analysis is supportive, but it is recommended to report both. Another case where sensitivity analysis may be needed is when patients receive non-protocol treatments before reaching the endpoint. So now we covered 4 of the estimates attributes, and we will move to the last one, population level summary, where the statistical analysis lives. I will move swiftly through this slide. It is just a list of some typical summary measurements we use. What is important is that even if in our CAT trial we stopped here with the difference in success rates, in real clinical trials, in the next step, we are generating proof that one drug is better than the other in form of a statistical test. Here is a quick reminder about statistical hypothesis testing. When a clinical trial proves that one drug is better than another, it is not a definite proof, which is not at all possible. What we do is probabilistic in nature. We claim that it is unlikely to get the observed treatment effect by chance when there is none, and reject the hypothesis of zero treatment effect. We need to define upfront what is considered unlikely. And a p-value, which a statistical test delivers, is a probability to observe the seen treatment effect by chance, although there is none. And the threshold for unlikely is the type 1 error, usually the 5% alpha we are all familiar with. When we plan a trial, we want to be pretty sure that we will be able to generate proof of an effect which really exists. To be able to do that, we need enough patience. We want enough power to reject the null hypothesis, and power is just the probability to reject the null hypothesis when the alternative is true. Power depends on the effect size and on the number of patients. So when we calculate the sample size for a study, the first two steps are obvious. Then you make assumptions for the control arm from the known data. Then you define your alternative hypothesis. You can use known data, for example, from phase 2 trial, if you have some, or you can decide to go for what would be considered as clinically meaningful difference. For the time to event endpoints, you will need a couple of additional things. By default, we assume that the sample size calculation is performed for one endpoint, the primary one. But what if we want to answer more than one question with the same trial? I have to justify the word efficient in the title of my talk. It is possible, but we have to be careful. If we just perform several comparisons, the type 1 error adds up. And as you see from the figure here, with more and more tests, the overall type 1 error gets closer and closer to 1. Adding questions to the trial is still possible, but it must be done under the control of the type 1 error, meaning that the overall probability of any false claim stays under your chosen alpha. Here are some ways to do that. You can split alpha. It is called Bonferroni correction. You can split alpha equally or not. Another way is hierarchical tests, where you don't even test the second hypothesis if the first one is not rejected. If you choose hierarchical testing, you should not report p-value for the second test if the first one was not positive. Unfortunately, it is not always the case. Further possibilities are improved Bonferroni, where you generate your p-values and you test the smaller one to the half alpha. And if it is significant, you can test the second one to the whole alpha. Or alpha recycling, where you first split your alpha, but then when one of the tests is significant, you can spare the alpha and pass it through and test the second p-value to the full level of alpha. Another very special type of answering to questions with one trial controlling for the type 1 error is switching between inferiority and superiority. If the non-inferiority margin is predefined, you cannot do it post hoc. It must be defined in the protocol. It is a closed test procedure because it is based on the same confidence interval, and it doesn't cost any additional alpha and no additional patients. The following figure is taken from the EMA guideline on the topic and illustrates the principle. So to show the superiority, the new drug is better, the confidence interval for the treatment effect must lie completely above 0. If it is not the case, as you see in the second, with the second confidence interval, you can look whether the confidence interval lies completely above your predefined non-inferiority margin, and then you can claim the non-inferiority. All methods described above can be combined. Important for the hierarchical test and alpha recycling, you need to think which objectives are more important or more likely to be positive, and you also have to respect the chronology. Do not put less important or late endpoints as gatekeepers. I put a nice tutorial in the literature list at the end of the talk. Sometimes you don't want to answer several questions, but you want to do several analyses for the same question. Why? You may test for high efficacy, and if you are lucky, you will read out earlier your trial. You plan for a moderate effect, but hope for big. A futility analysis can help you save resources and avoid exposure of more patients to inefficient treatments, if it is already clear that there is no hope of having a positive trial at the end. Both do cost in terms of sample size, because you need to control for alpha for the high efficacy. And if you plan an analysis for futility, there is some power loss and you have to adjust for that. Both can be combined with alpha control for multiple endpoints. And please be careful and don't plan your interim analysis too early, because, for example, if early events are intrinsically different, you may get a completely false signal. So think about the maturity of follow-up planning the interim analysis. And now let's move to the sample size calculation fine-tuning. If you are planning a complex trial, I suggest that you always start with a simple sample size calculation for your most important endpoint. Sometimes you see already here that it is not feasible and you have to rethink your population or your endpoint. And the next step would be think whether you need more complex assumptions. For example, non-exponential survival curves or non-uniform recruitment. Then plan for multiple comparisons and recalculate if necessary. And then plan for interim and recalculate if necessary. Interim analysis usually cost less than multiple endpoints in terms of patient numbers. An efficient trial is a trial which answers the question. An underpowered trial is always a problem. I listed some typical sources of problems. Try not to overestimate your event rates. Think about time dependence or non-proportional non-uniform hazards. For example, it is known that the early triple negative breast cancer patients, they have a peak in events in the second year and then it flattens out. Try not to underestimate your loss to follow-up rates. Think about non-uniform accrual, because the patients you put on the trial later contribute less to the power of the trial. If you plan a trial for the duration question, never ignore that patients are treated with the same treatment for some time. And you cannot expect any difference in that time. And the last one would be informative censoring pattern, which depends on treatment or disease. You need to think about it. For example, in an open trial comparing an active drug with observation, this could be a problem. Here we already come to the summary of the talk. A big part of the trial design is just common sense. Please use it extensively. Estimates language helps us to structure it. involve statisticians early on, start with a simple sample size calculation, and add complexity if necessary and feasible. Here is the literature list. And I thank you for the attention, and I want to thank all patients participating in our clinical trials and their families, and the team at the GBG headquarters with whom we are together on this journey.